Assalamualaikum and good day. My name is Nurina Damia binti Rifdi Takim from the Bachelor of Science Applied Microbiology program. Today, I will be presenting my Project 1 proposal titled Comparison of Microbial Contamination on between stainless steel and plastic PET reusable water bottles. This presentation will cover the research background, literature review, methodology, expected outcome and challenges, the Gantt chart, and reference. Reusable water bottles are widely used by university students because they are convenient and environmental friendly. However, these bottles are repeatedly refilled and often not cleaned properly, which may allow them to act as reservoir for microorganisms. These risks may vary depending on bottle materials, particularly stainless steel and PET plastic. Although students commonly use filtered or treated water, such as from cold machines, contamination can still occur after storage in the bottles. Repeated handling, refilling, and contact with the mouth can introduce bacteria into the water. Currently, there is limited local evidence comparing bacterial contamination between stainless steel and PET bottles among university students, which form the basis of this study. This study addressed three research questions. First, it determined the level of bacterial contamination in water stored in reusable stainless steel bottle compared with PET plastic bottle during typical daily use. Second, it investigated whether PET plastic bottle harbor high level of gram-negative bacteria than stainless steel bottles. Third, it investigated the prevalent and bacterial load of coliform bacteria and E. coli in both bottle types. Accordingly, the objectives are to compare total bacteria count between stainless steel and PET plastic bottle to characterize bacterial isolate from water and inner bottle surfaces and to detect and compare coliform and E. coli contamination. This study is significant as it increases awareness of drinking water safety among students and provides scientific evidence to support safer bottle choices in line with WHO drinking water safety priorities. The expected outcome is identification of which bottle material is more prone to microbial contamination and whether contamination occurs mainly due to handling and storage rather than the water source. Previous study has reported that continuous daily use of reusable bottles increases the likelihood of microbial contamination, especially when cleaning is inadequate. Some research also show the presence of coliform and heterotrophic bacteria at level exceeding safe limit. In comparison, stainless steel bottles generally harbor fewer bacteria than PET plastic bottles, likely due to differences in surface characteristics. Overall, bottle material plays an important role in microbial survival. As shown in the table, stainless steel bottles consistently show lower microbial contamination compared to PET plastic bottles. Stainless steel surfaces are smoother, more hydrophilic, and easier to clean and dry, reducing bacterial adherence. In contrast, PET plastic bottles are more porous and hydrophobic, retain moisture, and present a higher risk of bacterial attachment and biofilm formation. Overall, the literature strongly suggests that stainless steel is more resistant to microbial accumulation than plastic PET bottles. Now, let's move on to several factors influence bacterial adhesion or bacterial attachment on bottle surfaces. Firstly, surface roughness provides sheltered micronishes that protect bacteria from mechanical cleaning. Secondly, hydrophobicity and surface energy promote stronger bacterial attachment, particularly on plastic surface compared to stainless steel. Thirdly, the formation of conditioning film, which consists of organic residue from saliva or beverage, enhancing microbial attachment. Lastly, moisture retention and porosity support bacterial survival, especially in PET plastic bottles. Different types of bacteria has been reported in reusable water bottles, each indicating different contamination sources. Coliform bacteria indicate secondary contamination from handling and the surrounding environment. The presence of E. coli suggests possible fecal contamination, often linking to poor hygiene practices. Heterotrophic bacteria multiply during repeated storage and refilling of water bottles. Pseudomonas species are commonly associated with environmental and water system contamination. Meanwhile, Staphylococcus species are typically introduced through contact with the hand and mouth during bottle use. The workflow starts with selecting stainless steel and PET plastic bottle. Samples are cultured on selective media followed by colony counting to determine bacterial load. Morphological characterization is then performed to detect coliform bacteria and E. coli. In this study, a total of 40 reusable water bottles will be collected consisting of 20 stainless steel bottles and 20 PET plastic bottles. All bottles must have been in regular daily use by UiTM Shah Alam students for at least 1 month. Simple hygiene information such as how often the bottles are washed will be recorded only to describe contamination pattern. Both residual water and swab sample will be collected from each bottle. For water sampling, between 50 and 100 milliliters of residual water will be collected aseptically into sterile containers. For swab sampling, sterile cotton swab moistened with saline will be used. Swab will be taken from the inner wall, the cap, and the spout, which are considered high contact areas, and then transferred into sterile tubes. Serial dilution and spread plate techniques will be used to obtain countable colonies for the water samples. And for the swab samples, it will be directly inoculated onto selective and differential media, which are McConkey agar and eosinmethylene blue agar, or we call as EMB agar, for the detection of coliform bacteria and E. coli. Neutron agar will be used for total viable count, McConkey agar for coliform and gram-negative screening, and EMB agar for E. coli detection. All culture will be incubated at 37 degrees Celsius at 24 hours. And result will be expressed as CFU per milliliters. For bacterial identification, selective and differential media will be used. McConkey agar will be used to identify coliform and gram-negative bacteria based on lactose fermentation, while EMB agar will be used for the confirmation of E. coli, indicating by the presence of metallic green sheen. Enumeration will be carried out by selecting agar plate that contain between 30 and 300 colony, as this range provides statistically reliable result. The total bacterial load will be calculated and expressed as colony forming units per milliliters or CFU per milliliters using the standard SCFU formula. This allows quantitative comparison of contamination level between stainless steel and PET plastic bottles. Further identification will be supported by gram staining and microscopic examination to determine gram reaction, cell shape, and self-arrangement. This combined method allows both quantification and characterization of bacteria present in the water bottle samples. Contamination level between stainless steel and PET bottle will be then compared. For the expected outcomes, it is expected that reusable bottle will show measurable bacterial contamination. PET bottles are anticipating to harbor higher bacterial count and coliform or E. coli may be detected, indicating secondary contamination. Limitations include a small sample size and reliance on culture-based method. Handling contaminated sample also poses exposure risk to potential pathogens. For mitigation plan, all laboratory works will follow biosafety level 2 practices, including proper PPE, disinfection, and water management to minimize exposure risk. Strict sample handling procedure will minimize cross-contamination and ensure valid results. This Gantt chart outlines the timeline for sample collection, laboratory analysis, data interpretation, and report preparation. All references used in this study are listed here and are based on recent and relevant scientific literature. That's all from me. Thank you very much for your attention.